بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله، وأتمنى أن يكون الجميع بخير وعافية. I just want you to be more relaxed, the practical of today, and we have another practical, which is practical 7. Usually, even when the study is normal, we used to have what we call dry lab. That is, we introduced the practical, we make a PowerPoint show discussing the different slides and photographs of the parasites, and then you'll find those parasites demonstrated under the microscope. So we examine them by microscope. So even in your exam, the exam is going to be very simple. We'll give you a photograph of a parasite, for example, or an insect vector, and we'll ask you to identify. And then there may be a question or two relevant to that slide. So please just feel more relaxed and just study and make sure that if the picture is shown to you again, you will know whether it is a cyst or a trophozoite or a whole parasite or a larva, etc. So we have two practicals. The first practical, practical 6, it is about parasitic protozoa and arthropod vectors. And the other one is going to be mainly about the bigger parasite, the helminthes. Examples of nematodes, cystodes, and flukes with some of their vectors or intermediate host. So there is normal or very common laboratory techniques which are followed in a diagnostic laboratory, and for those usually the most important equipment is the microscope, because when you see the diagnostic stage of a parasite, whether this stage is a cyst, trophozoite, an ova, which is egg, or a larva, or the whole organism, then this is a precise identification. So the most important equipment in a diagnostic parasitology lab is the microscope. Occasionally, and for few parasites, we may need to culture them, which we call cultivation of parasites. And of course, there is serological tests, which are mainly identifying or looking for antibodies. And of course, as you know, the serological tests, they have their advantages as well as disadvantages. For example, if the antibody titer is less than the cutoff point identified by the manufacturer of the test, usually this may be a mild infection or borderline infection, or even just the scar of a cleaned or treated infection. In the laboratory, usually we can have two types or groups of specimens. One group is mainly for identification of parasite of the digestive system, for example. So usually it is examination of stool, maybe urine, and maybe sputum occasionally. And the other group is mainly for the blood and tissue parasites. So for examining a stool examination, for example, there should be good procedure of collecting fecal specimen. And in this case, it is preferable that the patient is not on antibiotics and not taking bismuth or any antidiarrheal preparation, no barium, for example, or mineral oil for the last, for example, week or 10 days. I'm saying this because taking these medications and preparations will interfere with the passage out of the parasitic diagnostic stages. In the laboratory, it is important to get well-collected specimens in clean containers which are well covered. So if you receive a specimen which is contaminated, for example, with water or urine, it is advisable to reject it and to request another specimen. And it is important to examine the stool as soon as it is in the laboratory. So ideally, the stool specimen should be examined within half an hour. Anyhow, it's not possible to examine all stool specimens within half an hour or an hour. So there is some acceptable methods that include, for example, to keep or hold the stool specimen for up to 2 hours at room temperature. It is also advisable you may refrigerate for up to 3 days, or to use preservative that can keep the parasite within the fecal specimen. If after stool examination the result is negative, no identification of parasite, if the patient, for example, is in the ward, it may be advisable, since the treating doctor is having high suspicion of a certain infection, is to request at least 3 samples, every other day 1 sample, not all of them in the same day. This will just possibly allow a period when the parasites are there in the stool, because parasites, they don't shed up their diagnostic stage regularly. So we spoke about preservative. We have at least 2 good preservatives for stool specimen. The first one is what we call polyphenol alcohol, and usually this preserves the trophozoite for permanent slides. Parasites with trophozoite like Entamoeba, Histolytica, and Giardia, Lambelia, etc. And there is methylate iodine formaldehyde, and you can use a concentration of 5 to 10%, and this preserves the Helminth X and the protozoan cyst, and it could be used for direct mounts and concentration. So examination of fecal specimen, of course, the simplest one, but it is not sensitive, is a direct wet mount. So precisely on a clean slide, you put 2, for example, drops of saline, that is physiological saline, or what we call normal saline, and then a small amount of 20 or maximum 40 microgram of stool, and then you mix this in the drop of saline, you put a covered slip, and then you examine, and this can help you to identify the motile or moving forms of trophozoites, if it is a hot or recently passed stool. Another way is to have what is known as a stained wet mount. That is, you have iodine, you take a small amount of stool, mix it up, put a cover slip, and you examine. The iodine will give a yellowish color to the specimen, and this makes it easy to identify the parasite. But we believe the gold standard test is to stain the specimen, but it is a longer procedure. To enhance the sensitivity of the test, you can use a bigger amount of stool. And in this, you can use what is known as sedimentation method, in which you take, for example, in a tube, which is about 15 milliliter. You take in the tube a bigger amount of stool. It could take almost a gram or even more. Then you add your solution, which is formalin ether, and you have to be careful. If ether is used, you have to keep it away from any flame, because it is easily inflammable. And in this case, you mix the stool specimen in the formalin ether, and then you spin it and you decant the supernatant, and then you can add another formalin ether. You also spin it up, and then you decant until you have a clear sedimentation at the bottom of the tube. Then you can take that sedimentation, put it on a slide, and then you dry the slide, and later on you have to stain. So it's a longer procedure, but it is enhancing the possibility of catching mild infections. Flotation, it is working on the principle between the specific gravity of the parasite stage and the concentration of your solution. And you can use what is known as zinc sulfate, or even if you don't have zinc sulfate, you may use the normal table salt, sodium chloride, by making a saturated solution. That is, you take water, for example, and you add salt, and you stir it until some of the salt did not dissolve. So you have a highly concentrated solution, and this is with comparatively heavy specific gravity. So also in another tube, of 15 ml tube, you can take 1 gram or more of stool. You fill it up with the zinc sulfate or sodium chloride sulfate, and you stir it up, mix it very well, and then you fill the tube until the solution comes to the rim or the edge of the tube, so that when you put a cover slip, it just rests on the solution. You keep it without disturbance for 10-15 minutes, so anything which is light will float and attach to the cover slip. So gently remove the cover slip, put on a cover slide, and you examine. So both sedimentation and flotation, they concentrate the or enhance the possibility of detecting mild infections. And of course, each one of these methods, it has the pros and cons, advantages and disadvantages. We said mainly the common specimens which they can usually come to the laboratory on daily basis, maybe the feces and the blood and urine, but I'm just going to show you a longer list which can come occasionally in the laboratory. I'm here giving precisely samples of the parasite. For example, in the feces, you can identify Entamoeba histolytica, GL Giardia lambelia, EV Enterobius vermicularis, TT Trichuris trichura, AL Ascaris lumbricoides, SS Strongyloides styrocoralis, Schistosoma mansoni SM, and Cryptosporidium parvum CP. So in the stool, you recover more. And there is, for example, hookworms, Ankylostoma duodenale, and Nicatra americanus. So a longer list of parasites could be diagnosed by seeing, for example, the cysts of Entamoeba histolytica and Giardia, as well as the trophozoites. And then you can see the ova, which is the egg of Enterobius, of Trichuris, of Ascaris, or the larvae of Strongyloides, or the eggs of Schistosoma mansoni, or the oocyst of Cryptosporidium parvum. In the blood, usually the most important parasite is Plasmodium, causing malaria. And for Plasmodium, there is 4 species which you know, which are Falciparum, Vivax, Ovale, and Malari. There is other minor species, no need for them now. B, this is known as Babesia microti. Usually it is a parasite affecting animals, transmitted by hard body ticks. However, human infection can happen. The parasite in the red blood cell resembles Plasmodium. Loa loa, we have a filarial worm known as loa loa. Sometimes, usually, it is limited in distribution, maybe in the forest areas of Central West Africa. And you have WB, Wuchereria bancrofti, causing the Bancroftian filariasis. Loa loa is transmitted by what is known as the deer fly or chrysops, while Wuchereria by different species of mosquitoes. The most important belong to the genus Culex, and in particular Culex fasciatus species. But in case of Wuchereria bancrofti, you need to collect night blood, because the microflare of the parasite, they come to the peripheral circulation usually at night. Loa loa, you take the blood. In urine, the common parasite is Trichomonas vaginalis. It is found in both male and female, and usually infection is through sexual intercourse. SH, Schistosoma hematobium. This causes infection by the penetration of the cercaria, usually emitted from snails, and usually they are found in water, these cercaria. And the parasite usually resides in the vesical veins around the urinary bladder, and the eggs usually are voided or passed out in urine. Wuchereria bancrofti, sometimes because it is in the lymphatic system, and the lymphatic system could be destroyed, and there is leakage of the lymph into the urinary bladder, so you have whitish urine, and it may contain the microflora of Escherichia bankrafti. In the sputum, parasites which are passing through the lung or living in the lung, larval stages, they can be diagnosed in the sputum and maybe occasionally in saliva. So Pneumocystis carini PC, Pneumocystis carini, causing an interstitial pneumonia, usually is diagnosed by examination of sputum. AL, Ascaris lumbricoides, SS, Strongyloides styrocoralis, and HWS, hookworms. We have two important hookworms, Ancylostoma duodenale and Necator americanus. So Ascaris, Strongyloides, and hookworms, they go through, during their development, they have what is known as tissue migration phase, and they go and live, and sometimes they molt as larvae in the lung, so they could be diagnosed in the sputum. Cerebrospinal fluid, usually parasite that invade the central nervous system, could be Toxoplasma gondii, which is very important in pregnancy. TR, standing for trypanosoma, causing ischemic sickness. And we said there is two species of trypanosoma causing human infection, Trypanosoma rhodesians and Trypanosoma gambians. There is what is known as free-living amoebae, but occasionally they become parasitic, and these belong to the genus Negleria. The most important species are Negleria fowleri and Negleria grueberi. Both people, they can get them if they swim in water which is containing this parasite, and then they can go through the nasal cavity, and from there they migrate to cause a severe meningoencephalitis, which would be fatal in a couple of days. AC, Acantha amoeba, and this is now a problem or causing many problems among people who use contact lenses. But the parasite also can cause a sort of meningitis. Bone marrow, Leishmania donovani causing visceral leishmaniasis or Kala-Azar can be diagnosed by aspiration of bone marrow. However, people, they should not go for invasive technique like collecting bone marrow aspirate, because it is not a safe or unnecessary. So you can examine blood. If it is negative, then you may need to go for more invasive technique like bone marrow. In the lymph gland, the parasite that lives in the lymphatic system is Escherichia bankrafti, the flareworm worm, Escherichia bankrafti, of course with other two species like Brugia malay and Brugia taimuri, but the most important one causing elephantiasis and very prevalent is Escherichia bankrafti causing Bancroftian filariasis. So they could be diagnosed by aspiration of lymph glands and to recover the snake-like structures known as microflare. In the liver aspirate, many parasites, they will find they go or live or their eggs are trapped in the liver. So you can find the eggs of Schistosoma mansoni, Schistosoma japonicum, Entamoeba histolytica trophozoite, Echinococcus granulosus, e.g. this is what is known as the dog tapeworm. Usually it is a small parasite consisting just of scolex and 3 segments, and usually people are infected when they ingest the egg of the parasite, and just develops into a larval stage known as hydatid disease. Leishmania donovani could also be found in the liver aspirate. So it means Leishmania donovani is very invasive parasite because it is infecting macrophages, so it could be found in different tissues and organs. Aspirina aspirate, you can also recover Leishmania donovani. Duodenal aspirate, Giardia lambelia, you can recover it from the duodenal aspirate as well as Strongyloides styrocoralis. And Strongyloides is the smallest nematode infecting man, and usually it's infected by penetration of the larva. In the skin, another parasite or another Leishmania, Leishmania tropica, causes cutaneous leishmaniasis. And also Onchocerca volvulus, which causes sarcomas or just nodules on bony areas, is and causing what is known as the river blindness. Then skin snips could be taken to identify Leishmania tropica, amastogotes, and the microflare of Onchocerca volvulus. Some parasites, they make or invade the muscles. For example, Trichinella spiralis. So people who eat pork, they could be infected with trachinella spiralis, which usually insist in the muscles. In old infections, or what we call chronic infection with Schistosoma sonnei, Schistosoma japonicum, and also Cryptosporidium parvum, rectal scrubbing may be helpful because in chronic infection with schistosomes, there is the tendency of the X to get trapped in the tissues. Bronchial aspirate pneumocystis carinii, causing pneumonia. This is now some slides to show you the parasite which you have recovered in the earlier lectures. So these are to show ring forms of Plasmodium falciparum causing the malignant type of malaria. Of course, for an expert person, he can identify, for example, the characteristics which are typical or more typical for falciparum. For example, if you find more or a couple of parasites in a red blood cell, this is suggestive of an infection with falciparum. And usually the infected red blood cell, as you can see here in the center, is not larger than the non-infected cells. To your right, you see a little bit magnified diagram where the ring forms, they are showing integration. It is divided into two. So this is what we call fragmented ring form. So when you find a ring form with fragmented nucleus, this is typical for Plasmodium falciparum infection because it's not enough to say this patient is having malaria. We wanted to know which species of Plasmodium is causing the infection because the regimen of treatment is different. Also, Plasmodium falciparum is having a different gametocyte shape and structure. And the gametocyte is the state which is when ingested by the mosquito will get the infection. And usually it ingests male and female gametocytes. So it is easy to know which is which. If you are very experienced, you see the nucleus, if it is fragmented or compact, then you know, yeah, but it doesn't make a difference. If you bring this slide, for example, in the exam, it is not enough to say this is a gametocyte of Plasmodium. No, you have to say gametocyte of Plasmodium falciparum because the shape is only typical for Plasmodium falciparum. If you see the cell here in the center, it is comparatively enlarged, bigger than the other cells. And also there is a lot of dots. These dots are known as Schaffner dots. So enlargement of an infected red blood cell and the presence of conspicuous or distinct red or pink dots is typical for infection with Plasmodium vivax. Of course, you could find milder dots in the case of infection with ovale, but ovale usually is the cell is somewhat oval and irregular at the poles of it. Also, for example, you can find a little bit of dots in red blood cells infected with Plasmodium falciparum, and those dots are known as Murr's dots. This is a schizon, but you don't see the cell membrane. And usually, in case of vivax, the number of merozoites from a mature schizon is about could be up to 24. The other ones, Bovali and Milleri, they have maybe 12 or up to 8, down to 8. However, in falciparum, the number is much bigger than this because it has a high fecundity of reproduction. And that's why it causes malignant malaria, as well as, of course, the infected red blood cells, they change the morphology, they become very rough, so they could be occluding or closing up very fine blood vessels in the brain, causing cerebral malaria. This is also a schizon of vivax, and if you could see the membrane of the cell is comparatively, it's not very clear here, but the cell is comparatively larger than the non-infected cells. And here it's a different staining technique method. If we brought this slide in the exam, usually it may be acceptable to say possibly this is a gametocyte, possibly of vivax, or even maybe ovale. However, it is just the cell is enlarged and there is clear dots, Schaffner dots, and the parasite or the gametocyte is almost filling the whole cell. And usually the parasite in case of malaria, they take a band almost in the center of the infected cell. Leishmania donovani, it is a group of parasites and showing different forms of infection. There is some that cause visceral leishmaniasis, which is known as Kala-Azar. There is other ones that can cause cutaneous leishmaniasis in the old and the new world. And the third one, usually parasite that cause damage to the mucus-containing surfaces like the nose, etc. So we have what is known as mucocutaneous leishmaniasis. In this parasite, if you look to the diagram to your right, it shows the nucleus and kinutoplast. So the parasite is somewhat oval or rounded in structure, and there is no free flagellum. That's why the name is known as amastogot. Or generally we say LD bodies, Leishmania donovani. L for Leishmania and D for donovani, known as Leishmania donovani. So all of those small structures are having the same oval or somewhat rounded structure. There is no free flagellum. Trypanosoma gambians and Trypanosoma rhodesians morphologically are indistinguishable, but both of them are intercellular parasites, and all of them they look somewhat wavy. Of course, Trypanosoma cruzi a little bit different because most of the fixed specimen they don't have this very wavy structure. They may be assuming an L or a C-shaped structure with comparatively large nucleus, and of course during the multiplication of cruzi, some of them they could be intercellular or intracellular. When they are intracellular, they can destroy the muscles and they can cause such infections like megacolon and megaesophagus, etc. But the African species, Gambians and Rhodesians, they cause a sleeping sickness, and usually it may be some people they suffer from chronic infection, while one of them could be acute, which is fatal within a year or less. Toxoplasma gondii usually is a very important infection during pregnancy, and it is more dangerous if it is an infection at the early months or days of pregnancy. It can lead to abortion and other formation, for example, of stillbirth, brain involvement, etc. So the parasite recovered is usually very simple, somewhat oval or crescent in structure. The central part is more or less just showing a dark, large nucleus. Pneumocystis carini that can cause pneumonia. Usually, to see the oocyst of this parasite, you need a special stain, and here we have what is known as silver stain. And usually, it is just cup-shaped cyst, not much to see inside, maybe just something known as sporozoites. Entamoeba histolytica, it has two stages, the cyst and the trophozoite. So the disease or the dysentery is mainly caused by the activity of the trophozoite. If you see this photograph, you have a larger and a smaller form. That's why the size varies from 10 to 30 or even a little bit more than 30 micrometer, depending on how young or how big or old the trophozoite is. But one characteristic which is helpful to identify Entamoeba histolytica is the nucleus. If you look to this nucleus, the nucleolus is, or what we call endosome, is in the center of the nucleus. And if you look to the margin of the nucleus, the membrane, the chromatin is comparatively smoothly distributed on the margin. So this is characteristic of Entamoeba histolytica trophozoite, because there is another species known as coli, but the nucleus is not in the center. We call it off center, and the chromatin, the peripheral chromatin, is not regularly distributed. It's irregularly distributed. It is very easy when you see a trophozoite which is ingesting red blood cell. For sure, this is a trophozoite of Entamoeba histolytica, because just to see a trophozoite with the characteristic nucleus, it may be histolytica or it may be the trophozoite of another indistinguishable species, which is known as Entamoeba dispar. So Entamoeba dispar, morphologically indistinguished from Entamoeba histolytica in shape. But when you find In gastric blood cells, it is typically, without hesitation, this is a trophozoite of Histolytica, the disease-causing form. Here you see also two forms of Giardia. To your left, there's two specimens. These are trophozoite, the actively feeding stages, which have a concave structure on the anterior ventral side by which they attach, or cup-shaped structure by which they attach to the intestinal villi. And each trophozoite is having two clear nuclei. And of course, with the central axostyle, from where also there is 4 pairs of flagella, it gives it the shape of a face. And some people, they say it is a smiling face, although I don't see any smile here. To your right, you'll see the infectious stage, which is the cyst. And a mature infectious cyst contains 4 nuclei because it is a trophozoite. When conditions are unfavorable, it starts to contract and to be able in structure, withdrawing all the flagella inside and forming resistant cyst. And each of the nuclei, the 2 nuclei, divides, and so we end up with 4 nuclei, which is an infectious cyst. Trichomonas vaginalis, it exists only as trophozoite because transmission here is direct. There is no need for a resistant cyst because usually most of the infections are during sexual intercourse. So the parasite, it doesn't have a typical shape, and we call it amoeboid. The amoeba, it doesn't have a proper shape. So here you could see at least there is 4 flagella from one end. Just under them, there is a 5th flagellum which is attached to the body of the parasite, forming undulating membrane. So the 4 flagella and the undulating membrane can help the parasite to move or to jump. So usually it has a jerky movement. If you look up, there is a large nucleus, and also there is an axostyle extending across the body. Cryptosporidium parva, usually the oocyst, could only be seen by using what is known as acid-fast stain or uramine stain. And usually it could be pinkish thread in structure. So the oocyst may be containing 4 sporozoites. The insects of medical importance are Anopheles mosquito, which transmit the Plasmodium parasites causing malaria. And this, as you see here, is a greedy mosquito which is feeding taking a lot of blood, so it's almost dripping out without well digesting the blood it took. This is a diagram to show you the developmental stages of a mosquito. So usually a flying insect can lay eggs. Here the eggs are laid in group, or what we call egg raft. This is typical for Culex mosquitoes. The eggs are laid in rafts, while Anopheles, it lays single eggs, and each egg is having lateral a little bit of flotation structures. The eggs in fresh water, usually they hatch to release actively feeding stages and moving stages. These are the larvae, and usually they click to the surface of water by their siphon to take atmosphere air, oxygen. After a couple of days, the larva will pupate. So we have a dormant pupa, which after a couple of days will give you the flying insect. So for control of such mosquitoes, you may use insecticides for the flying insects, and you can use what's known as larvicides, which are chemical against the larvae, which is the feeding stage. So control could be, for mosquitoes, could be by molluscociding, that's insecticides, or larvicides, and sometimes maybe by biological. You add to the environment, for example, fishes or snails that can feed on the eggs and the larvae and pupae of those mosquitoes. In Africa, the sleeping sickness is transmitted by the Glossina, what is known as tsetse fly. It is comparatively a big, colorful fly, as you see here. In South America, Chagas disease, which is caused by Trypanosoma cruzi, is transmitted in the feces of these bugs, which are known as Rodnius bugs or radiophyte bug. So usually, when people are sleeping, this bug, especially people sleeping in cracked containing huts or shabby huts, so these are good environment for the breeding of those bugs. So at night, they can just dwell over the surface. As you see, they have very strong proposes. So when they're taking blood, the uh, injury is a little bit irritating, so they defecate, and the parasite is found in the feces of the bug, so they could be pushed in the small lesion left by the biting, or if it is near the eye, it can go in the eye, causing glaucoma. And of course, from there, they can migrate to invade, for example, other organs, causing megaesophagus or megacolon and heart problem as well. Phlebotomers or sandflies are tiny flies. Comparatively, they look very hairy and with long, and usually these they breed in soil-soaked, water-soaked soil. So usually it's not easy to identify where they breed. So that's why they are named sandfly, because they breed in moist containing sand. There is what's known as itch mite, Sarcoptes scabi, and this causes a sort of dermatitis known as scabies, which is بالعربي الدارجة يا شباب is known as الجرب. Usually, scabies is more common among animals, not on human. So usually the itch mite is having clumpy, short limbs and some hair structures. So this insect or mite, it goes through the soft skin between, for example, the fingers on feet or any soft skin, and there they lay eggs, and the eggs they hatch to give the nymph, and the nymph they keep crawling, so they cause sometimes a very annoying dermatitis, which is known as scabies, our jarab. You remember we mentioned another parasite which looks in the red blood cell, it looks like a plasmodium parasite. This is babesia. So babesia is transmitted by what is known as exodus, which are hard ticks. Ticks could be divided into two groups, soft body ticks and hard body ticks. And most of the parasites which we see attaching to the animals, either in the ear or at the udder or the soft skin, are hard ticks. And these ticks, depending on the species, they could carry viruses or cause infection by viruses or bacteria, or also parasite. If you look to the diagram on your left, you can see that there is a dark, tough structure limited to the thorax of the tick. This is known as scutum, and the scutum is not stretchable. And then the body, for example, of the female tick is very inflatable, very soft, leathery. It could be expanded so that the parasite, I mean the insect or the tick, can take a lot of blood, because these female ticks, they need blood to produce plenty of eggs. Here you see this colorful tick. The dorsal side, it looks like a shield. This is the scutum, which is strong, not stretchable, and that's why the male takes only comparatively less blood, because the abdomen is not stretchable, as you could see in this photograph. You see here, this is a female hard tick. The scutum is very limited to the anterior end, and you can see this very big inflated abdomen with a lot of blood. This is what we call engorged tick. Thank you very much. This brings me to the end of the first practical, which is practical 6. Please do visit those slides so that you make sure if you see any one of them in the exam, you will be able to identify what is it, and then you can answer one or two relevant questions to that slide. Stay safe and well. Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh.